I'd like to welcome everybody to our second presentations and orientation for our National Science Foundation funded research traineeship on harnessing the data revolution for stakeholder-guided environmental science. We shorten that down to stages. So I'll go through the agenda very quickly. We're going to start this morning with Dr. Murgulet welcoming everybody, and then we will go into our trainees, Cohort 2 trainees presentations on their capstone projects. We'll break for lunch, which will be here in this room, and reconvene in the afternoon for longer introductions and orientation for Cohort 3 trainees. Hopefully we will be ready to break no later than 4 p.m. So with that, I will hand it over to Dr. Murgulet.
Good morning, everyone. Thank you so much for joining us for our second stakeholder graduate student trainees presentations and afternoon orientation. And welcome to Stages, which Stages is short for Stakeholder-Guided Environmental Science, which is our research traineeship program funded by the National Science Foundation. And it's what the goal of the National Science Foundation, NRT. Sorry, I'm getting very distracted.
Sorry.
Are we good? Okay. To support interdisciplinary and evidence-based research, traineeship for graduate students that will advance the graduate students and including master's and doctoral degree programs to pursue careers in STEM. Before I go further, I would like to acknowledge our upper administration present here and their support. And I'll start with Dr. Madi, Executive Vice President of Research. Dr. Madi, thank you for being here this morning. Dr. Rudowski, our Provost, Vice President of Academic Affairs. Dr. Janet Donaldson, our Associate Vice President of Research. And I know they have very, very busy schedules, and I wanted to make sure that we thank them for being here and for their support before they have to go. And I know they're going and coming back. But thank you very much for being here. So before going into a little bit what the program is about, I would like to just present what our program, what's the organization and management of our program. Of course, we work with, like we just introduced our upper administration here and our Dean of the College of Science, but I am the program director, and then I work with an executive team. Dr. Jennifer Epley Sanders is our new faculty that joined the program as in the executive team as a co-PI that replaced Dr. Jinbing Zhu that moved on to another institution. And we thank him for all of his support. He's now serving in a role of the advisory board. And then Dr. Liu, Chintao Liu. Chintao, I don't know where you are. I see you there, yes. Dr. Pollock, Jennifer Pollock in the back, and Dr. Tissot in the back. We have senior advisors and advisory committee. We have Dr. Douglas that just opened the remarks here, she is our program coordinator, program manager, and we owe her a great deal of thanks because we couldn't do this without her. And then we're working with an external evaluator, Mr. Scott Berg, who is annually coming down and interviewing our trainees and our upper administration. He also interviews our stakeholders as we're evaluating the program and we're trying to improve from year to year. And we also have faculty mentors that work with and support our trainees. So introductions will be happening during the orientation time, but as we go through this earlier morning, I just want to again here's our team with this program, and I already introduced everybody. So there's always that phase of the proposal, right? You as a team, and we have to thank the research office for investing time and money and in trusting us to be able to do something so great. So, you know, as a team coming together and being coached by the research office, former vice president, associate vice president of research, we have talked about what we would like to see in our programs and what we think our graduate students could benefit from training. And as a matter of fact, just make it more formal, some of the aspects of the Coastal Marine System Science program that were there, but we weren't making those efforts, right? So we were supposed to be very interdisciplinary, but it's very difficult to do interdisciplinary work because everybody has their own program, their own niche. And then two, we are working with a lot of state, federal agencies and communities, but not necessarily relating back to them what we can do and what our graduate students can do. So we proposed to address several challenges. So one of them, understanding the interrelatedness of the critical challenges facing coastal areas like climate variability and impacts like degrading water quality. And so with that interdisciplinary and convergent approaches to engage in communication with stakeholders and policymakers and answer the relevant questions for them. And to represent students from different backgrounds, include them into this traineeship in environmental and data science, and create this new pipeline from coastal communities to the environmental and data science workforce. So our program addresses all these challenges to create a new generation of diverse environmental scientists and coastal and marine system science that will be prepared to conduct big data analysis and visualization and to use deep learning like artificial intelligence and machine learning techniques to team research and co-developed with regional stakeholders. It's a lot in there. It's packed, right? But it is including all of those needs, challenges. So our goals and objectives were to prepare cohorts from STEM graduate trainees for leadership in interdisciplinary team science settings with an understanding of coastal and marine systems and applicable tools of data science to equip them with communication, stakeholder collaboration, and other professional skills necessary to succeed across workforce sectors, to foster the development of a network of partners engaged in making the Gulf Coast more resilient to climate change by co-developing research questions pertinent to regional work and needs, and working with these stakeholders to address the questions and problems and increasing the utilization of public data sets. To establish and disseminate transformative STEM training, research, and evaluation advances in coastal and marine system science that produce a sustainable and scalable graduate education model. So, packed together, it's kind of what our program model is there. I will not go through that, but move to what we've done. Just very, very briefly. So as part of the Coastal Marine Systems class, which is supposed to be a interdisciplinary coursework that will introduce our students to the different disciplines that come together to, to resolve those complex issues that I've talked about. We added to the course a field trip where we're taking students to the headwaters of a river and come along, follow it down to the coast where it's discharging into our bays or estuaries. And so the first field trip was in the first cohort that we had along the Nieces River watershed. And then the second one was along the Colorado River. So we've learned that with the first cohort that it was pretty difficult to do that along the Nieces River, a lot of private property. And it just happened we were in a dry year and a lot of dry spots. However, we also learned that if there wasn't groundwater flowing in those spots, the river wasn't flowing either, or we did see it where it was coming off the bank somewhere further downstream. So it touched that groundwater that you actually don't see, which is my passion. So we put together, like we said, a community of partners that we engaged with, and so far we have about 12 that have engaged with us. Among those, as you can see, NOAA, Texas Water Development Board, Texas Parks and Wildlife, the Texas General Land Office, the Sea Grant, and so on, coastal and-based estuaries. And we have the workshops that take place at the end of the spring semester where we bring everybody together, the stakeholders, the students and their mentors, and discuss which projects the students are going to engage as a group to based on the data that the stakeholders have presented as being available and the discussions about what questions they need to be answered. And so this is where we establish those groups and those research questions, and from here on the students work on the projects throughout the summer as part of a new course, the Data Blitz. And in fall, as part of another course, the environmental forecasting. So here are our, so far, 17 trainees. We have had 17 trainees from 4 different programs, 9 master students, 8 PhD students, and there have been 5 projects, so group projects. From these trainees, we have had 4 master students that graduated and 1 PhD that just defended this semester. And from there, and 1 advanced to candidacy. From there, 4 out of those 5, they have jobs, very prestigious jobs, and they have been hired by some of our stakeholders or those that have heard about the program. And we're hearing back from them as this program actually was, might have been one of the reasons they got that job. So the first cohort, we had a precipitation group project and the seagrass. If anybody from the first cohort is here and would like to be, you might be, what is B? Yes, B is there, and she came back this year as a mentor to our cohort 2. And then our second cohort, which we'll present to you today, we have the Shorebirds Group, that just chose the short names, the manta rays group and the bacteria group. So the Shorebirds worked on a project developed, was to answer a research question developed with the Coastal and Bays Estuaries program. The manta rays worked on a project developed with NOAA. And the bacteria group worked with Mr. Pinchback from Texas General Land Office, who is in attendance here today. And we also have, I believe, NOAA in attendance online. So what other things we did? Professional development. So as I mentioned before, our external evaluation is there to guide us on how to progress. It was a new program, although we were building on an existing program. It was a lot more than as just continuing with the practices we had so far. So we have received feedback from the external evaluator on what, the stakeholders might have seen as maybe improvements, or our trainees are being interviewed once during the time they're trainees, but there are surveys as well. So Cohort 1 had monthly topics for seminars, science communication series, alumni panel, resume and CV workshop, and collaboration and teamwork. And moving into Cohort 2, we added weekly seminars, as there wasn't enough that we packed in monthly. We just didn't have enough of the professional development topics that we could include. So again, weekly topics: team science and project management, communication and collaboration outside your team, communication style, science communication, which included research progress presentations and group project progress presentations. They had to every month present once on both of this. Experimental design, faculty, stakeholder-driven research, peer review and publishing, and the alumni panel. And we thank the alumni panel, our alumni is coming back and talking to our graduate students, as we also thank our stakeholders who have engaged with us earlier this year with the second cohort and came and presented and engaged in conversations leading into that May workshop. The students already knew what they wanted to do and what was available there, pretty much. So the other accomplishment, so what we've done, we have now a trainee certificate, which is also called Stages, but we changed the name to Society Targeted Approach in Graduate Environmental Science. It started in January 2025. I think so far I signed 2 or 3 degree plans that have added this to their, it's a secondary degree, my understanding. So you get your master's or a PhD and you get this certificate added there. It combines 12 credit hour coursework, which is pretty much this, the coursework that we had as part of this traineeship. But we added the 3 credit hour of stakeholder-guided research. So in addition to that coursework, the student that wants to add the certificate to their transcript will have to engage with the stakeholder during a semester for a 3-hour research. So, and it doesn't have to be, it's not outside their research, ideally. They would just want to get feedback on their topic of research and potentially, pivoting a little bit to come out with an answer that's relevant to the community or the specific stakeholder. And we have a new scholarship that goes to support a trainee that goes through this certificate program, the Patrick and Julie Nye Scholarship. So we thank our stakeholder partner for coming forward and helping with this. We have awarded it, I believe, twice so far. And so we will do the same for this next year. So what's next and what's needed? As you go through this and you are realizing, go from the proposal phase into where you're just, I want the funding, or this is exciting, we want to do this, and then it comes, and then you see how much work that is. And if this program wouldn't allow us to have a program coordinator on the budget, not sure this would have been possible. That's to say that, which is at the bottom, but should be at the top, to continue this program beyond funding, we would need a dedicated person that can coordinate all of these events. So it's time for us now to prepare what's beyond this program. We have a new cohort coming in, but it's also our last cohort. And with that in mind, our NSF, the funding agencies, the agency would like to see from us is that we institutionalize some of the elements of this program. And so we have already added a certificate and the course, the summer data blitz, where it's actually pretty popular now because students outside the program are engaging in that course because they have access to faculty that can guide them with data analysis of their own research. So we didn't have a course in summer where students could just progress on their research before. But we need other we need to institutionalize others. So partner workshops, like I said, it's time commitment. Somebody would have to be in charge with that and have the time to communicate with stakeholders to put together those workshops. And there's also funding for that, right? That you have to have a little bit of funding for that. Dedicated faculty to teach the artificial intelligence, machine learning coursework. We had Dr. Tissot for the longest time, put a lot of time and effort into teaching these courses. But it's very difficult for him to engage with when he is doing research, and that this is time commitment, and we need more than the environmental forecasting. We have to have someone available in summer to coordinate the big data blitz. And from the trainees' feedback, they think that an introductory course would be very beneficial before going into the more advanced environmental forecasting. So they're more aware of where the data is, what kind of models are available, what should they look at going into the more advanced coursework. So they're more successful. So that requires a faculty that could teach those courses and be dedicated to. And then engaging with faculty from other colleges to maybe help us with the soft skills seminars. The soft skills is something that came up repeatedly, I think, from stakeholders and our alumni that work at federal or state agencies that say that everything else you can teach them there that they need to know in their jobs, but the soft skills are the ones that they really need their new employee to have. So these are just a few of those what's next and what we need. All right, I think I got to where we handed to our cohort 2 for presentations, and is this the order, Audrey? Okay, so we'll have first the Manta Ray group. Guys, it's your time.
Thank you. Okay, that's okay. Should we get started? Good morning, everyone. We are the Monitoring Race for Improved NOAA Enforcement, or the MRINE Group. And this is our final presentation. I'm Mahima.
I'm Mandy.
I'm Remy.
So we'll go ahead and introduce ourselves. So as I mentioned, my name is Mahima. I'm a second-year master's student studying marine biology here at Texas A&M University-Corpus Christi. And the goal of my research is essentially to understand the ecology and diversity of marine microbes within the Gulf of Mexico.
My name is Mandy, and my research involves examining the sulfur metabolomics, genotypes, and size diversity of Paludilli seagrass.
My name is Remy. I work in computational chemistry as well as bioinformatics, trying to understand the effects of toxicants on model organisms. And I'll pass it on to Mahima.
Thanks, Remy. So before we get started on getting into the nitty-gritty of what we did this past year, we wanted to go ahead and introduce our study subject of the giant manta ray, known as Mobula birostris scientifically, and just kind of present why it's such an important organism and such a key organism to our project. So as you might guess, the giant manta ray truly does live up to its name, with a wingspan of up to 26 feet and a weight of up to 5,300 pounds. These are highly migratory fish, and they also feed on planktonic and micronectonic organisms, and they have been known to kind of migrate to where their prey species are.
But again, there are a lot of knowledge gaps on the depth and the capacity in which they migrate. They do have a lifespan of up to 45 years, which is quite impressive. I didn't know this, with the low fecundity, essentially meaning that they aren't giving birth often and regenerating their population as much as maybe other marine organisms might be. And this all plays into the fact that they are classified as threatened under the Endangered Species Act, known as the ESA. Their main threat is commercial fishing, where they're usually caught as bycatch, but they're also not immune to being threatened by other anthropogenic activities, whether that's oil and fishing activities or anything else, and of course, climate change, as we all know. So moving on, in case anybody's really interested in the scientific classification of the Mobula virostris, I have that up there. The thing that I did want to bring everybody's attention to is, I should do it over here. They have these unique black spots under their underbelly, and the patterns are unique for each individual manta ray, and this could come into use when trying to identify individuals. But generally, they are these really magnificent and wonderful organisms, and it has been a really huge pleasure to be able to study them. But moving on to their geological range, they do have a really wide range as evidenced by this map. And this is significant because we know for a fact that they're highly migratory, but this map kind of shows us that these aren't organisms that tend to be specific in their geographical range, making it more difficult to identify where kind of their key breeding grounds or nursery grounds might be. And this plays into the two main knowledge gaps that our project attempted to focus on. So the first one was better decomposing the migratory patterns of the giant manta rays. So as I mentioned, these are highly migratory species, but we aren't fully aware of the depth and the breadth of where they're migrating, how that looks seasonally and temporally. And we also want to better understand what the environmental drivers of the patterns and distributions of these species are. And these knowledge gaps alone do have a lot of significant impacts. So for one, not understanding the human impacts of climate change impacts our understanding on how we can better protect the giant manta rays. So it's really important to have a better understanding of that. If we also don't really understand what their migratory patterns look like and where they maybe spend more time giving birth and have nursing grounds, we can't properly delineate critical habitats and nursery grounds in order to better conserve them. So we're really not being the most effective in carrying out these conservation strategies. And the last implication of this knowledge gap is that we aren't really able to put forth the most efficient conservation efforts. And again, given that these species are considered as threatened under the ESA, we want to make sure that the conservation efforts we're putting out are as efficient as possible. And right now, that's just not happening. So again, filling these knowledge gaps is absolutely critical so we can better protect these species. And I'm going to go ahead and pass it on to Remy. He's going to talk a little bit about the deep learning.
All right, well, earlier on, we've heard about a lot of different names such as artificial intelligence, machine learning, and deep learning. And so before we dive into the model itself and what we've done, I want to first highlight the differences between these three different model types. And essentially, we have artificial intelligence, that's kind of the big all-encompassing terminology for machine learning and deep learning. And what distinguishes these different models is how they're trained and how and what type of data they're trained on. So for example, we could have a random forest. This would be, I believe, a machine learning approach, whereas a deep learning approach would be something like a convolutional neural network. And this is exactly at what layer we're working on in this particular project. We're working with convolutional neural networks. So instead of AI, we'll be using the terminology deep learning. And so that's what that means. And so now I'll pass it on to Mandy.
Okay, and so our plan for the project was to train a deep learning model using the YOLO V8 program and PyTorch to identify manta rays from aerial and satellite images. So we can get kind of a massive overview of where manta rays are in the entire global sense instead of having to look at targeted places and go doing a physical survey. The purpose is to track the movement patterns, possibly identify individual mantas from those dark spots Mahima talked about earlier, but not quite there yet. We want to do this to confirm or reject previous location prediction models. The map on the right is from a paper written by one of our stakeholders. They compiled a bunch of sightings data, and dotted them on a map to show where manta rays are statistically most likely to be found. And we want to make sure that is that actually the case, or were there just more people there that time? And so our final products are going to be a Linux-based AI model, deep learning model for them to run, and creating an SOP for the MRINE program and YOLO V8 training protocols so they can modify them if they want to. They can add different data and they can change it to look for anything else that they want to. And so this is just a roadmap. I'm going to go in depth on everything here, but this is kind of how our year went. We started with the idea of using Maxar satellite images. That didn't end up working. Then we went to using Google Earth images. That didn't end up working. And we finally ended up using aerial survey pictures from NOAA. And so first we need to narrow down what time we want to look at and where we want to look. NOAA told us to look in Florida, so that part was easy. When to look, we compiled all of their sightings data, made a histogram of the months, and determined that the top 3 months to look are February, March, and April. And so our first approach, the Maxar satellite images. So NOAA had an archive of Maxar, which is a third-party company. They had an archive of these images. We requested images from our latitude and longitude range during those top 3 months, but that ended up not working out because the resolution wasn't quite high enough. They were 14 to 17 meters per pixel, whereas a manta ray is max 8 meters, so it was hard to make a definitive identification from that. So then we moved to Google Earth Engine, which are open access free satellite images. We used the Sentinel-2 satellite. Once again, we filtered the images down, we put them into regions, look in those top 3 months, and then you had to manually search through those images. And this is what an Excel sheet looked like. I made a list of all the images Sentinel-2 had during that time frame, during that region, made a list of all the images, so then we could go look through them. There was over 200, which is in all a good problem to have, but it does take a very long time to scan through all of those images, zoom in enough to be able to see a manta ray, and then the resolution was again not quite high enough. So then I'll pass it on to Remy to say what we did.
All right, so for solving this issue about having satellite resolution imagery, we then asked our stakeholder, well, what would you like us to do? And they had actually access to aerial imagery, which is basically manta rays imaged from either a drone or a helicopter or plane. And so we then proposed the idea that maybe we take these images, train the models, but modify them such that we naturally decrease the resolution to what we would expect to observe from a satellite vantage point of view. And so in order to do this, we take this image and then we lower the resolution. So we take the average of the neighboring pixels. And for this code, we need to do several things before then. First of all, we have to label the object that we want to detect, which is this manta ray right here. And then we also wanted to expand the number of images that we could train our model upon, because we had in total around 45 images or 46. And this is not just enough to train a model. We need to have more images. Approximately 100 images is what is recommended for optimal training. And so to get around this issue, we decided to also alter some other parameters, which Mahima will be discussing here in the next figure.
Thank you, Remy. So yes, part of what I worked on was essentially manipulating our original raw aerial photos or aerial images to try to make our data set more robust. And I did that essentially by manually changing the color palette of all of the images of manta rays. So I'll kind of walk us through what I did here. So to the left, we have a raw image of a manta ray, and then to the right, I went ahead and annotated that as an object using the computer vision annotation tool called CVAT. And essentially, we created a label for the manta ray, and we just decided to call it Mobula FPP. We saw a couple of different species of the manta ray, but we were a little bit hesitant on definitively identifying them, as that's not really quite where our expertise lies, and the resolution was just not optimal enough to make that sort of definitive conclusion. So this was the first layer of annotation that occurred. But then as Remy mentioned, we wanted to make this data set more robust, utilizing some of the techniques that we learned. And so what I did was I really just went in and changed the colors by hand. This is a process that I would like to try to automate just to make things more robust. Luckily, like Remy mentioned, our data set was not big enough at this point for this to be a huge impediment, but essentially what I did was, drive up the blue saturation, maybe lower brightness, and essentially make the image look quite different in its color composition. And then once I was, happy with what the end product looked like, and I felt like it was sufficiently different, I repeated the process of annotating it on CVAT, and I labeled it as the same object. And this is again the same process for another raw image. So raw image, annotation via CVAT. And then I've gone on the bottom and edited the colors here. And actually in some of the backgrounds of our slides, you'll see some more of these really wonderful aerial photos of the manta rays so that we can kind of display some of these really wonderful images. But in terms of scaling down the images to make them optimal for satellite training data, I'll pass it over to Remy and he'll talk a bit about that.
All right, so to tackle this problem, essentially what we wanted is a before and after image with our before aerial image and with our satellite imagery resolution. And so to determine what would be the proximal pixel resolution, we referred to a single image that our stakeholder provided, and using a standard software, we were able to use the calibration curve, the calibration scaling to determine what is the pixel resolution for that image, and then try to repeat that resolution in the after imagery. And so the way by which this was done is using this script right here, in which this parameter right here is modified such that we can get that targeted pixel resolution. Now, for the deep learning approach of the training process, there are several things that we have to take into consideration. But before we get into that, I want to ask you a very simple question, which is, what number is this up here? What number is this? 9, right? And of course, what a stupid question. But to us, it's very simple, and in a very streamlined fashion. We don't even have to think about it. We see the number 9 and we're like, oh, that's the number 9. But if we want to get a machine to do the exact same thing, we have to break down this process into several steps, right? So if I ask the question, well, what makes this a 9? Then that's a very deep and profound question, and we have to take this number and take it apart. So with a 9, I see a loop up top. Sorry, I'm going to point over here. Because I'm going to break my neck. So up here we have a loop up top, and then we have a line that goes down. So that is what makes a 9. So if we had a loop up top, and then let's say instead of a line at the bottom, we had a loop at the bottom, what would that be? That would be an 8. And so in a deep learning model, we and specifically convolutional neural network, we have this input layer and this output layer. But in between, we have what is called the hidden layer. And what this hidden layer does is exactly what I've described here. It takes apart the image and makes a relationship between the pixels. And so as you can see here, the inputs activate nodes. These nodes activate later nodes in the hidden layer. And here we have 784 pixels, and then we see that process, right? And then the node 7 will activate. And if we try another number, let's say number 2, same thing, different nodes get activated, leading to the image of the 2. Now, for training, there were 3 major rounds of training after playing around a bit with the YOLO V8, which is the platform that we use for the training. It's made by Ultralytics, and actually was one of the first image analysis platforms for deep learning. I think it began in 2012, which was at the beginning of the boom of artificial intelligence and deep learning. And so by the way in which this process works is that we take our annotated images that Mahima provided, and then we also have some blanks in there, and then we give the model the labels as to what we're trying to annotate. And then in the script for training the model, there are several parameters that we have to take into consideration. So YOLO V8 is very advantageous in the fact that it breaks down an image into little boxes, right? So this is the size parameter that you see right here. And so that's the length and width for an image. And so that allows us to then plug in any other type of image that we would like. So no matter the width or the height of the image in terms of pixels, we can use that image. Now, other parameters that are important are the EEP. Okay, I'm not going to use that screen anymore. So can I take this off? Okay, because I feel all right, so there we go. So we also have other parameters like the epoch. Now the epoch means how many times the image is used during the training. So it can recycle through the images and modify the weights for the different nodes that you saw being activated. And then we also have the device, which is 0. 0 stands for GPU. This was trained on my own computer because for this type of model, using a graphics user processor is much more advantageous. If you use something else, you might break your computer, which is what I've done. And so then you also have then the batch. The batch is another parameter that we have to take into consideration, but 8 is the normal standard parameter. And so with the different layers that we have, we can also change how many of those layers are present. So we can have a small model, a medium model, and an extra large. There are other intermediate model sizes that we can choose from YOLO V8, but those are the 3 that we're choosing. So that's what we're using for the first training approach. For the second training approach, however, what we can do also is take a pre-trained model, in this case a model trained on identifying ships from satellite imagery, and then modify it such that we use the same data, but instead of training it to identify boats, we can then train it to identify our object of interest, which is the manta rays. Now, why would we want to do this? Well, because the weights are already pre-trained to distinguish what is ocean and what is an object of interest. And so that gives us an advantage when we have such a small amount of data that we want to train it on. And then there was a third round of training as well, in which Dr. Chuso gave an idea that we would extend the number of epochs from 200 to say 1,000, and so that was also done. Now, after we identified those parameters and we ran and trained the models, we get also some data that we can use to determine whether or not our model is precise, and then we can use that data to also compare how our models are effective between one another. So the main parameter I'm going to be focusing on is fitness, which encompasses the other parameters. The other parameters are all about: is the model effective at identifying the manta rays? Is it confusing the manta ray with the ocean and other parameters? Is it completely missing them? Or there are other types of errors that can be thought of. And so these are parameters effectively measure that. And from the fitness, which is a score, we can then make our comparison. So in total, there were 11 models trained individually with different epochs of training, different sizes of the models. And looking at the fitness, what you can see is the one that is closest to the number 1 is the one at the bottom, which was the one in which we're retraining the model to identify not the boats, but the manta rays. And I'll talk to you as to perhaps why that is in the results section. Now, for the results, I'll be using also this convolutional matrix, and then essentially what I would like you to take into consideration is how dark this color is up here in this box. The darker this color, the more effective it is. The darker these boxes are, the less effective it is, and the more errors are present. All right, so for the smallest model, Nano, trained at 200 epochs. We can see some darkness here, but as well as some confusion right here. What you see over here as well is these are not the okay, we're back. So these are not the labels that actually Mahima put, but this is the actual model giving us a prediction that and identifying that as a manta ray. So that's pretty cool. And then so for our medium-sized model, the results are very similar in terms of the convolutional matrix. But when we get to the largest model, the one with the most hidden layers, with the most nodes, we see a huge drop in the efficacy of this model. And so my question was, well, why? Why is this the case? Well, and the most likely answer to this is, well, we didn't have enough images. So the more nodes you have, perhaps, yeah, the better that model might be at distinguishing differences between different and recognizing patterns like what I've described before. But there's so many weights that need to be modified that we need more images to modify and to tweak those weights, which was not the case because we ended up with around 74 images for the training. Now, when we retrained the model, the boat model, this actually had the best performance. As you can see, we have the darkest up at the top and some light shading blue on the other sides. And in terms of the accuracy, it was the best. It was overall an 85% accuracy. Now, when I tried to retrain the model with more than 200 epochs, the idea was that as we retrain the model, the weights were going to get more and more fine-tuned. But YOLO V8 Ultralytics, the engineers who designed this platform, also provided a optimization code such that if the layers are not being tweaked or aren't changing, then it's going to stop the training because there's no point in wasting battery time for training a model that's already met its peak performance. And so that's what I found. Actually, at around 100 epochs of training, the model was already optimized for the data set that was provided. And so with this, we then developed an SOP for our stakeholder to utilize, as well as stakeholder 3 if you guys find yourself in an image analysis problem. And this SOP is designed for anybody who wants to use YOLO V8 from start to finish. The only thing that's required is a basic knowledge of how to use the command line. And there are also some instructions for changing these some of these scripts that I've displayed up on the screen. But really, you can only, if you have a basic understanding of coding, you should be able to do exactly what we've done. And so, like Mandy talked about also, we developed a program that can be used directly in the command line. You don't have to open up Jupyter Notebook or know anything about Python. You can put in just your path to your image set that you want to analyze, and then get an output with an Excel spreadsheet, but as well as some annotations on the actual images. And so with that, I'll pass it on to Mandy.
Okay, so while it was fantastic and we got a pretty high accuracy, there are definitely limitations to what we did. Chief among them being we did not have a whole lot of images to train it on. We had less than 100 images, which is less than ideal. And also we had to use those same images to validate it. So that can definitely cause a bias because it could just be recognizing that image had a manta ray because I recognize that image, not that I recognize the manta ray. So that's a definite issue that could be a definite bias. And then there was a lack of spatial diversity. All of the aerial images were taken along the East Coast. Mahima altering the colors was trying to supplement for that, trying to make it look like it could be taken in Galveston, making it a little more brown, could be taken in Flower Gardens, making it a little more light blue, you know, but it's always better to have actual images taken from different places. And there was also a lack of diversity in the manta ray's behavior. All of them were they're perfect, like flat out, you know, you could see all the wings, you could see all the shapes. None of them were upside down, none of them were twisted a little bit, so that is also a limitation. And with that, I'll hand it on to Mahima to give our acknowledgements.
So we wanted to go ahead and thank Stages, of course, for giving us the opportunity to even get to meet our stakeholders and learn so many valuable techniques on machine learning, data handling, statistical techniques, you name it. We wanted to also go ahead and thank our wonderful stakeholders at NOAA for providing us with this project to even begin with. I know we were all super excited about it the second we heard about it. As you can tell from our backgrounds, this was reasonably different than what we do, and it presented a really great opportunity for personal and technical growth. And of course, we would be remiss without thanking Texas A&M University-Corpus Christi and NSF for funding. And with that, we will go ahead and open up the floor for questions. Thank you. I'm gonna, oh, yes.
Are you aware of any other databases, maybe European or Asian, that may have had?
Are you aware of any other databases, maybe from Europe or Asia, South America, wherever, that may have imagery of manta rays? It looks like their distribution is pretty broad across the globe.
Yeah, at the moment, I am not aware of any. I'm sure they do exist with other
I think that's a good question.
Yes, yeah. I don't doubt that there are different organizations that probably have access to those data sets, but primarily as far as we're aware with our scope of what we did with NOAA, we didn't work with any images outside of the United States, but I think that that would be a really good approach to, again, like supplement our smaller data set, because I don't doubt that, I mean, if people know how threatened they are, I would find it odd if there weren't another repository that existed, but we didn't explore that.
That's a very high detection rate you presented. And have you thought about introducing some noises, like putting very quite sharp into the image?
So what do you define as noise? Could, like you said, another object like a shark? In our data set, we actually did have sharks. I think we had n equals to 2 sharks.
And then there was
N equals to 3 sharks, but we also had humans because these were drones. We had an image with a boat in there as well. And so, yeah, we had a diverse amount of noise, not to a greater degree that in terms of seagrass. Well, did we have seagrass in it? I believe so.
Sargassum.
Some sargassum. And yeah, so we had that type of noise. But what would be interesting in other, if we had a bigger data set, what I would be interested in terms of noise would be waves. I think that would be very interesting, but we didn't have access to that. So it's a very good question.
Yeah, in the image that we did have of the boat, when I was going in and kind of manually editing the images, I actually tried to edit out the boat, but in turn ended up creating another object. And I think I still ended up putting that back into the training data just so that there was something there. But yeah, I mean, I think that your question definitely represents the need for, again, a larger set of images, because I think that noise just makes the model more robust and kind of identify images.
Hello. So I actually have a little bit of a background in plasma science and have worked with a lot of people that do drone surveys and have done them myself. I'm curious if you could recruit citizens for this project to provide you with drone footage of manta rays. I know there's a lot of people in Florida as well as in other areas of the world, I see, who have a lot of like personal footage of them, and it's on Instagram and things like that. So I'm curious if you could factor that in to give you maybe more behaviors or other areas of the world that you could look at.
Yeah, absolutely. I'm sure there's tons of people, because I mean, you see a manta ray, you're gonna take a picture of it, right? But I think what it was is there was a time restraint. We didn't have enough time to set up that much of an outreach. If you guys pick it up, that's definitely something you could do. But I think that would be very helpful to have more. And our model also can be fed directly into drone feed. You could put the video in, it'll identify it.
And I also think in the paper with the image sightings that Mandy had presented way back at the beginning of her slides. It's okay, if anybody wants to see it, I'll go back, but I think some of the sightings that they found in there were from citizen scientists or people who had seen manta rays, so there definitely is a capacity to incorporate that, and I think that would be a really great angle for outreach for sure, so thank you. Should we check online to see?
Nick, you had your hand raised. Would you like to unmute and ask the question? Yeah, I threw it into the online chat if you guys are not able to hear me.
I think he said he typed it in.
We might have to read it from the chat.
Yeah.
So Dr. Nick Farmer, one of our NOAA partners, said, excellent presentation and impressive work despite the unanticipated challenges we faced with image acquisition. What resolution of satellite imagery do you think would be the minimum for useful, greater than 80% accuracy, model performance? Do you think the aerial survey training images will be sufficient for satellite imagery? Or do you think even with the downgrading of image resolution, the goal would be to acquire an inventory of satellite images for training? Thank you all again for your hard work.
Very good question. So we could expand it with the aerial imagery. The thing I think we discussed on that was that, well, satellite imagery has different cameras types, different wavelengths that it'll detect, and the light also bouncing off the manta is getting filtered through the atmosphere, so the wavelengths and the colors will be slightly different. So ideally, if you knew how those colors would be changed going from the aerial to the satellite, then yes, theoretically that'd be possible. As to the resolution, 30 centimeter pixel, I think that's the best I've found in terms of Google as to what type of satellite you would need. Perhaps you'd be able to get away with 60 centimeter per pixel. Do you guys have any other inputs? If that answers your question.
Thank you.
Is there any other questions?
We're a little ahead of schedule, so while we get the next presentation set up, we have a 10-minute break.
Okay, our second students presenting today are our Shorebirds Group for short, and I will let them introduce themselves.
Okay, good morning, everyone. My name is Allison Gerard. I'm in my second year of my PhD program here at TAMU-CC in the Coastal and Marine System Science program.
Hello, everyone. My name is Justin. I am also getting a CMSS PhD, and we work together on this project here called Identifying Inland Wetland Habitats in Rural Southeast Texas for Migratory Shorebirds along the Flyway.
And we were working with our primary community partner, the Coastal Bend-based and Estuaries program, and specifically the bird program within this organization.
Okay, so sorry about that. CBEP has had a long-term goal of being able to inform and incentivize wetland management practices in the context of being able to increase protection efforts and how we can support migratory bird foraging habitat when we need it most. And so we propose to draw a project, to have a project drawing on some of the inspirations of similar programs where we have incentivization of farmers and landowners to manage the land in such a way that can support these migratory birds. So one of these examples is this bird returns program that is primarily done in Central California. And so this is a program that provides some financial compensation as well as some guidance and consulting to help these farmers kind of create these pop-up wetlands that can be available for these birds to provide the resources they need when they need it. So when these migratory birds are going to be around in those specific areas. And so these programs have been built on a lot of kind of on the ground research. They've also used a handful of kind of citizen science approaches where they're having people on the ground, taking pictures of birds, identifying what areas could be the most important. And after a few years of development, they were able to kind of get this program running and has seen some great success since. And so after seeing a lot of this great success in the California Bird Returns program, one of the things that CBEP wants to consider is can we kind of make a similar program a reality in Texas? And so that was kind of the overall goal here is how can we get closer to being able to inform wetland management strategies. And so the Texas coast, especially kind of where we're at in South Texas, is a very important area for a lot of these migratory birds. So it's a part of what we call the Central Flyway. The Central Flyway is this big kind of bird highway that connects the northern hemisphere to the southern hemisphere. So you have these major migratory patterns going on, where you have birds kind of living in really, really cold regions in the summer, flying down here for the nice weather in the winter, and using the resources here within these wetlands to kind of support the energy needs to make these long journeys or be able to nurse and support the populations. And so these wetland habitats are really, really important for these birds and they're highly dependent on them. However, as you can imagine, land use change has greatly impacted how much of these wetland habitats are available or when these wetland habitats are available. And so we want to see if we can find some opportunity to support these migratory birds through some deliberate land management practices. So our study area is going to be not too far from home here. We have an area somewhat close to Houston, and this is an area that is known for a lot of its rice farming. It is a pretty kind of wet area, and they have a handful of areas that, with proper management, could support a large amount of these birds that are naturally coming through here year by year. And so we sought to use some imagery of this area and decided to pick this area because it is kind of a system that we're a bit familiar with. And so there was supposed to be an image here on the right panel that seemed to have disappeared during getting this PowerPoint loaded on here. But this was just kind of highlighting, it was supposed to be just an aerial image of our study area. From this NAPE imagery. And so this data source was from the National Agricultural Imagery Program, where we have this small planes flying over every year during the growing season, combining all of these images together, filtering out kind of tally days to try to kind of come up with a nice, clean, processed product that we can then analyze and use pretty easily. And so our project objective was to create an image classification model to help characterize these palustrine wetland habitats, these inland wetland habitats, and with a bit of a focus in the agriculture regions along the Central Flyway, as oftentimes these agriculture regions are going to be areas that are highly affected by these land use changes. And so we wanted to do so with high resolution imagery, as high resolution imagery does provide us a level of detail that can allow us to classify these habitats in a lot more detail than we would be able to otherwise if we're using some, let's say, 10-meter resolution imagery. And so here on the right is an example of what this NAPE imagery looks like along the river here, and we can see a handful of these different wetland habitats very closely either around or within these farmed habitats. And so we had some anticipated goals here, and then the first thing that we wanted to do is we wanted to classify and identify these different types of wetland habitats. So we first wanted to be able to say what type of habitat are we looking at here, and then also be able to say how much of this habitat is available. And so if we do kind of implement these land use changes down the line, we'd be able to quantify the effects there or project areas that are going to be really important for us to look at. And overall, our results are going to serve as like a proof of concept. And so we want to see how accurate, how precise of a model can we make in terms of the class and the subclass levels of these identifications, and what could be possible with high resolution imagery to kind of hint at what could be possible once we start getting more kind of better data to work with in terms of higher temporal resolution down the line.
See if I like this in my hand. So yeah, as Justin was saying, we specifically wanted to work with the native imagery because of this really high resolution that it has. It's really going to be your only freely accessible imagery that has better than 1 meter resolution in terms of spatial on the ground resolution. And we feel like that was really important for our objective of trying to classify wetlands specifically in a palustrade system. These inland systems that don't always stick out as much as you might see recognize wetlands in an estuarine system. So I'm going to sort of talk about the data a little bit and the workflow that we used throughout the year to obtain these objectives. And I'll start first with just some details on the data download and the preparation. So the native imagery is freely available, and we accessed a large chunk of that from the Google Earth Engine repository. And once you sort of access your area of interest, which we based on, this rural area in Texas, where our community partner is actively doing some monitoring on the ground. So once we had our data of interest, we're able to load that into a GIS software such as ArcGIS and do some annotations there. So we used a sort of combined workflow where we started in ArcGIS and then took our imagery out and put it through some Python codes to go ahead and train our model and move forward with our objectives. And so the GIS software makes it really easy for us to annotate and load in our different classes. And that was a big part of the project as well, is just trying to familiarize ourselves with these types of wetlands and try to get an idea of what we could realistically characterize just with imagery alone. So once we had the annotations, we could go ahead and use some of the GIS tools to rasterize the annotations and match them up with our actual imagery. So we could eventually put this into a convolutional neural network type of model. We're going to be using the UNet model. And once we have that sort of loaded into the GIS environment, we're able to split that imagery and the masks that are associated with the imagery into these neat little tiles that we can then feed into our model. And then within the model that we are building in Python, we're able to sort of optimize the different parameters of our convolutional neural network and try to evaluate the model with validation data and then transform that to the test data and actually come up with our results. So some details of sort of how this workflow went. Again, we're loading in the imagery into ArcGIS so that we can sort of easily visualize it and define our different classes in a feature class and make these different representative annotations for each class. And then using some geoprocessing tools, we're able to split both the imagery and the raster layers that have our annotations into these different tiles, and we can sort of neatly organize them into directories that are able to be fed into the model. And then once we have the data sorted, it's just sort of a matter of coming up with computing resources and the time to go ahead and run your model and test different parameters to get the best results. And then eventually, once you sort of run your model on your test data and you extract the different annotations and you make the different predictions for your entire imagery set, you can then load that back into the GIS software for visualization and then using other sorts of geoprocessing tools for some of our statistics and so forth. So I'm just going to talk a little bit about the architecture that we decided to use for our model, which is this UNet architecture. And it's been used previously in different types of segmentation tasks, and it's been used previously in things like biomedical imaging for detecting things like tumors. And so this is a pixel-wise segmentation where you are assigning masks to your imagery, and each pixel essentially is going to end up with its own value. And the model works through these sort of different layers, like Remy was talking about in the MRINE project, where we essentially start with all of our information. We sort of downsample that to catch the lowest resolution features that are available, and then we sort of work our way back up. And as the model starts to encounter more details, it revisits the imagery to try to assign classes to the different pixels. So what we end up with is these sort of segmentation maps that match the imagery that we put in, so that every pixel in the imagery is matched with a pixel in your feature class. And that's what we can start to feed into the model. So then a big part of this is trying to define our different classes. And me and Justin both happen to be analytical chemists and not biologists or wetland ecologists. So, this was sort of difficult. It seems like these classes are pretty serious. They take their wetland classifications very seriously, where you have a system and a subsystem and a class, different water regimes, and potentially special modifiers that you use. And again, we're just trying to look at imagery alone. We're not out there on the ground and we don't have the expertise. So we didn't want to stretch ourselves too thin and make assumptions that we could back up. But we thought through investigating the imagery that there were some different features that we would be able to start picking out. So for example, it's pretty apparent in the high resolution imagery when you have a shrub-scrub type of vegetation dominating versus an emergent wetland with sort of these marsh and fen type of vegetation. You can also start to get an idea sort of the water regimes, right? So if we had really advanced this model, then you might start to incorporate things like soil moisture data and other data sets that are derived from various different measurement techniques. But we essentially are just trying to say, does this look like it's underwater all the time? Does this look like it might get inundated some of the time? And then some of these wetland classifications depend on the substrate type. So we're talking size of the particles. Is it mud? Is it sand? Is it gravel? And obviously we're not going to be able to achieve that with imagery alone. So we're sort of just going for the low-hanging fruit, looking at unconsolidated grounds. Basically, does it look muddy? Does it look sandy? And then because we're targeting an agricultural area, and that's really sort of the overarching goal, is how can we reach out to farmers and landowners, we're sort of honing in on the most agricultural type of classes. So these are the Capstone subclasses that we eventually sought to characterize. So again, all of these are going to be inland in the Palestine system, and we sort of just made our best guess with these different water regimes based on what we could visually sort of parse out from the imagery. So a lot of these unconsolidated shores, again, that just has to do with areas that are sparsely or unvegetated and having these different water regimes. And it's pretty evident through the imagery when you have things like aquatic beds, where you have standing water, deep water habitats that might be covered with some surface vegetation. And then we tried our best to pick out some of the emergent wetlands and the shrub, scrub, and forested wetlands, because those are sort of natural areas that are probably going to be attracting wildlife anyway. So when we look at some of the unconsolidated shore, especially with the artificially flooded and farmed special modifiers, those might be the areas that, if we can identify them, we might be able to, eventually develop some sort of program where we're targeting those sorts of areas, right? Where our community partner can reach out and say, hey, you have this land that could potentially be used for this purpose of helping the birds. So you start to get an idea of where we can target the approach. And so Justin's gonna go through some of the examples so you can see how good this high resolution imagery is for classifying some of these different systems.
Okay, so we're gonna go through some of these examples, and before we do, I just want to bring it back. What we're working with is this imagery to where each pixel is just going to be like a small table associated with, right? So we have like a red, green, blue value. There's a number associated with that, so we don't get the amount of detail that we can get from physically being there. And so wanted to show the examples of what these different classes look like with the name imagery themselves. And so we're going to start with the aquatic beds. And these aquatic beds are going to be, you can think of them like ponds. Ponds where you're constantly having water there, and you're going to be having some growth, like plant growth growing within them that is going to be like aquatic plant life. So not like flooded corn or something, but like what we see here in this top image right here. So we have within aquatic beds, we can have a variety of what these systems look like, whether we have different kind of watercolors due to different kind of water chemistry that we have going on there, or we can have different types of plant cover too. And so we can see sometimes we have this kind of bright green vegetative material, where other times it's kind of this very light patchy material. The next class that I wanted to point out is these emergent wetlands. So these emergent wetlands can sometimes look pretty similar, especially if we happen to have the image of an emergent wetland while it's flooded to these aquatic beds. So these are areas where we have evidence of transient water. Maybe it's not always flooded, but after kind of a large rain event or something, water will pool there and stay there for a while. We usually have vegetation there that is going to be able to survive on dry land, but does okay when it gets flooded underwater. So think of classic herbaceous vegetation with roots. And so some examples here from the nape imagery from above, we can kind of see this dark green, this light green characteristic in these plants that give us a good clue that this is an area that usually can hold some water throughout the growing season. The next class that we wanted to show examples of is the scrub shrub wetlands. These scrub shrub wetlands are going to be wetlands where we're having woody plants growing. So instead of what we see in the emergent wetlands, where maybe it's like grasses and small little herbaceous plants, we're starting to see some more woody stuff that maybe can be tall as me or a little bit taller. There is a delineation between scrub shrub and forested wetlands where we have kind of trees that are about 20 feet tall. Now, since we physically can't tell how tall these trees are just from this nape imagery, we didn't kind of go through the efforts of delineating those two, although we are in South Texas, so we don't have too many large forested areas, especially as we come farther down towards us. But we were able to see some cool examples here where we were able to see kind of evidence of this wetland habitats kind of between these trees and shrubs here. So the identification for these types of wetland is actually going to be probably pretty dependent on the tree cover. But hopefully we can kind of get some info of the water as well. The next major class that we wanted to be able to identify is unconsolidated bottoms. And as Allison showed not so long ago, we did break that unconsolidated bottom down a bit. So unconsolidated bottoms are going to be areas that are also prone to hold standing water, but are going to have a different type of cover to them. So these, the emergent wetland, we saw a bit more of these terrestrial plants getting flooded. These unconsolidated bottoms, you can think about them as kind of more barren areas where we're having a lot more like different types of sediments and different geology there. And so we can commonly find examples of this in like flooded fields. And so if you've driven past a cornfield or any other type of field, you can usually find some sort of kind of area where the water naturally pools. So from the imagery, what that might look like is kind of barren spots where the crops don't grow well because they've been flooded too frequently, or you have different types of vegetation growing there or different growth patterns that can kind of highlight where these areas are. And so once we have kind of looked through these classes and based off of what classes we're seeing present in our study area, then it was the task of annotating them. And so we would go through and physically just kind of point out what each of these classes that we're seeing are. And so here we have an example of these masks that were created to kind of cover what each of these what we classify each of these areas to be. And over to
Okay, so we got a model and we ran it and it made some stuff. It doesn't look as pretty. I was hoping to have a really colorful map that was just gonna be beautiful and show you all of the wetlands in our study area. But we have some line graphs. So we basically started with, you know, the obtaining the data and annotating the data was a great success. Actually, ArcGIS, after some learning, is a really functional tool and eventually user-friendly if the user is open to it. So we were able to sort of get some results and check back in and then go ahead and make some adjustments to our model. So I started here with this initial model on the left, just using some sort of default parameters from an existing UNet model. Again, this is a popular model, sort of easy to access existing models, but then of course you want to go through and optimize your different parameters for the task at hand. So what I was seeing with the initial model was not very great performance. We have these high loss values that weren't really plateauing over a long amount of time. And so coming back in and sort of adjusting the model, you can mess with all sorts of things here. So you can change things like the learning rate or simplifying that decoder, right? So how many layers does your initial set of pixels go through before it is minimized to that low resolution? You can adjust all those sorts of settings. And then you're also going to be playing around with the time that it takes you to get those results as well. So as it turned out, when I sort of changed some of these settings, we ended up with that really pretty line graph where we see the loss minimizing, the training and the validation loss in agreement with each other, sort of plateauing at that low value over about 48 months. And so that was the model that I ultimately saved. Once the model is ready, you've already gone through, you've trained it, you've gone through your validation data, and now you just have this big data set that you're waiting to actually test. And that was the part that I was hoping to have in a pretty map for you. We're almost there. Check back in with me next week when it's Christmas time, and I'm gonna have this ready, and I'm gonna show Santa, and it's gonna be fantastic. But the model worked, it did stuff, and it's just not quite at the point where I can visualize it yet and do the sort of summary statistics that I would like to. Because eventually, you know, we want to make use of this. We want to say what area is unconsolidated bottom, artificially flooded. We want to be able to characterize and quantify these different wetland areas so that we can use this as a tool, something that could be informative in the future. So we got off to a really good start, and we learned a lot, but I don't have the pretty map to show you yet. But we certainly can talk about the challenges that we face and the future directions that we could take or our community partner could take, maybe in conjunction with members of Cohort 3. And we have to address the obvious limitations of our approach, even if everything worked out really well. So we're making some major assumptions here about these different wetland classes. And again, these people are very specific about how they assign their wetland classes. So if the pebbles are more than 0.25 centimeters in diameter, you can't call it gravel, you have to call it whatever, right? So that's sort of the level that this classification system is used at, and we're not going to be able to achieve that with imagery alone. If you really wanted to make use of the imagery for an entire classification schema, you'd probably want to have some ground-based data and probably some other remote sensing data as well when it comes to identifying the different water regimes and, making inferences about soil moisture. Because there are existing soil moisture models out there, and we explored some of that data for our project. These models are really complex, and they're not going to have this type of resolution that we were hoping to achieve on this really small scale, especially in a complex farm system. So again, these aren't your typical coastal wetlands where everyone thinks, oh, they're so pretty, let's protect them. These are farmers who are more concerned about what they have to do with their properties. And so a unique challenge here for these palustrine human modified types of areas is finding out where can we best optimize the land and manage it in a way that is productive for both landowners and for the birds. And then we have some other challenges as well. Justin's going to talk a little bit about one of the goals we originally set out to achieve that wasn't quite possible with the products that we had.
And so, as Allison suggested, some of our initial goals was to really hone in on some of the temporal variation that we see in a lot of these habitats, right? And so, whether we can visually see a wetland there or not, can oftentimes be really dependent on the type of year, or maybe just the day or the hour that the image is taken. How frequent after a rain were we able to see this area? Maybe we can't see the area because there's cloud cover on the days that are most likely to have the most moisture. And so, since we use this named imagery, which we selected due to the very high spatial resolution of the products, we did kind of make the sacrifice on the temporal resolution there. And so that named imagery is that one snapshot of the entire growing season. So it's kind of a bunch of planes passing over, and they're stitching these images together. And so you can have one side of your study area from 1 month, maybe the other side is from another month. And so that is kind of this one thing that we just couldn't get around with the type of data we're working with. But any progress we can make with this data could easily be applied towards newer forms of data. So if a landowner or project, these projects get taken up, and maybe they're flying their own drones or their own kind of aerials, ways to collect data, then we can easily apply those as well. And so, being able to capture that temporal certainty is going to be really important, especially when we're kind of considering it in the context of these wetland, in these migrations, right? And so these birds are oftentimes going to be needing these wetlands to be around during, let's say, March and April, or kind of the early part of the year, let's say early spring, to then August to winter or so. And so being able to see what these wetland habitats are looking like, whether they're there and available for the birds or not during these times, is going to be the most important. And so that is kind of the overall goals that we'd like to be able to achieve if given the chance to. And so I think one of the biggest challenges to then take it to the next level is going to be being able to get these data sources that have a combination of high spatial resolution and high temporal resolution. But we believe it was most important to kind of focus on the high spatial resolution first, kind of see what level of classification can we get to with the wetlands. And then if we can prove that we can classify these wetlands, we can identify these key habitats, we can then kind of make a better case to invest more resources into some higher temporal resolution data products.
So with that, we'd like to thank our community partner, Coastal Band-Based Estuaries Program, and the STATUS program, the program faculty and staff, and everything that they do, and our peers in the cohort. It's a great time. But yeah, it was a lot of fun learning lots of things, and we were very happy to take any questions. Thank you so much.
Wonderful job, guys. I just had a quick question about your map that you had that is not quite done. So on the bottom, I see there's a difference than the top. Can you explain what that difference is?
So I tried to prove that I was actually doing something. Okay, so the part that I'm at now is that I have all of the predicted masks. So you run the model, and then the output is, assigning these different pixel values based on what your model predicted or was trained on. So trying to load it into the GIS, I have a grid that sort of matches up with my study area, and I have all of the little cells that each tile is supposed to go into. And the green is just overlaid over my raster file of the study area. And the little gray boxes are the output tiles that I got from my model. So I basically just put it there to show my model did something, it produced something, but I'm not quite sure what's going on with that. I tried to mess with some of the attributes and changing some of the values, but it wasn't quite where I needed it to be. If anyone has any idea, feel free to let me know.
Yeah, I just had a question about the native imagery. Is it just visible light, or did y'all have access to infrared as well?
That is a great question. So they oftentimes have the infrared imagery, the infrared channel available as well. That is something that is very kind of region specific and year specific as well. And so ideally, if we could use that, that would give us a lot of info in terms of what kind of vegetation we're looking at, and could give us a bit more info that we just wouldn't be able to see with 3 channels.
Great presentation. I just wanted to ask a question about, so you were able to sort of go through some processes to refine the model performance. Are you at the place, or were you able to tell, are there certain, habitat types, for example, that your model is having a hard time predicting accurately, or is it like the edges of the image, or do you know where if you had a magic wand, the places are that still need tweaking to be relayed where you'd like it to be?
So I imagine maybe a lot of the issues, like I said, it went out and ran, so it's hard to say what is actually holding it up now. I tried, in the annotations, we tried to sort of ignore the obvious class imbalance of it mostly being farmland, so it's easy enough to go and highlight all of the farmed areas because there's so much of it. So I was trying not to just excessively choose those areas. Something that we were running into in we actually kind of redid the annotations in masses a few times, because what was happening is the whole tile would be like a mass, because you would just have a big farm plot, and then the tile size was the size of that farm plot, and so then the mass was just all black. So I think that was causing some issues, and I don't know if it would have been better if we would have tried to take a more sparse annotation approach, where we're just taking little bits of the farmland. I think also too with the forested and the shrub scrub, I mean, that is such an obvious and distinctive feature on the imagery that I think that might be causing some issues. If we can actually go ahead and come up with summary statistics, I think there might be some issue with the class imbalance there.
And maybe a little something to add to that. And so Allison hinted at, we did take a few steps before landing where we're at. And some of those was kind of getting an idea for how similar these classes might be. So some of it being just visually, if I look at can I really tell what's the differences here? But we also ran a few sport vector machine models that we didn't present here today with different classes. And one of the things that we saw in that too was large amounts of misidentification, especially once we start adding in more classes. Like if I try to identify a road or a building on top of identifying all these wetland habitats, started getting a lot of mis-IDs. And so on top of some of the classes just visually looking very similar, maybe having some overlap, I think kind of honing in specifically to our question, instead of trying to annotate and classify everything, just classify what we care about, is what we get.
Hello, my question is, how many images did you annotate? And then for the class imbalance, like, is there a ratio that you could give, like, oh, we have 80% farmland, maybe 10% ponds?
So I basically I had the whole TIFF, and then I'm just annotating it randomly, and then when it splits into tiles, if there is a mask associated with it, it gets output into its own directory, and that becomes the training data. And so that ended up being just under 700 tiles. And then I split it like 80-20, and then I tried 60-40 for the training and the validation. But really, it was just a limitation of me not wanting to draw any more polygons. You know, I probably could have done a lot more, and then, you know, tried to get at that. As for the actual statistics on the class imbalance, I don't have concrete numbers for you. I just was trying as I went. Like, oh, I've been circling farms for a long time. Let me move on to something else. And try to just eyeball, you know, in that attribute table, how those were adding up. But I never actually went through and, you know, got the exact proportions.
Okay. And then for this picture, did you look and see if there was like any hidden layers that weren't showing up at the moment, but digging deeper, maybe that's what's going on. So I've ran into similar issues with transcriptomic data.
Yeah, this was a little bit of a frenzy. So when we were like putting the final product together, whatever I tried to do at first started loading each individual predicted mask in as its own layer. So then I had like 5,000 layers in my map, and it wouldn't stop. And I just had to force close everything. So I didn't get to investigate it too much. That got to me a little bit. And then we had to choose our battles. Yeah. Come over the presentation for you guys.
Thank you.
Yeah.
Maybe I can add something to that too really quickly. So we did investigate a little bit of what looking at the IR band included looked like. And so with that, it was pretty cool. Although we didn't proceed with the IR band all the way through to the end, we were able to see some of these ponds where maybe the RGB values, the water looks like the exact same color. But then all of a sudden you have the IR band in there, and one is kind of glowing a certain color just based on how we decide to visualize it, and the other one's not glowing. So that could be like an interesting avenue that really looks clear.
Okay, thank you, Justin and Allison. Okay, our next group is our bacteria group. So I'll invite Laura, Quincy, and Claudia for their presentation.
Hello everyone, like I said, we are the bacteria group, but we like to call ourselves EnteroWatch because we are working with the TGLO Texas Beach Watch, and we are identifying the key drivers of Enterococcus levels in Texas coastal water using machine learning techniques. My name is Laura Button, and I'm a master's student in the Coastal Marine System Science program. I study groundwater contamination and unincorporated rural communities, and my advisor is Dr. Dorina Murgulet. What I did primarily on this team was mapping and data analysis, and I am from Ski, Texas.
All right, my name is Quincy, and I am a master's student in the CMSS program as well. My background is in atmospheric science, where I use ground-based and spaceborne radar to understand the properties of extreme weather events. And my advisor is Dr. Liu, and my role on this team was primarily in computer programming and Python and data analysis, and I am from San Antonio, Texas.
Hi everyone, my name is Ifeanyi. I am a PhD student in the Coastal Marine Science program. My research is centered on coastal hydrology and cartography and transport. My advisor is Dorina Murgulet, and my role on the team is writing and analysis. I am from Owerri State, Nigeria. So I'll go ahead and give us the outline of representation of today. It runs from relevant information down to the references. And so, since you're working on Enterococcus, Enterococcus is a gram-positive bacteria that is an indicator of topical contamination. And so, sources of these Enterococcus have been linked to leaking septic systems, sewage discharge, stormwater runoff, and wastewater treatment plants effluents. All right, so this is just a picture showing us the different sources of these Enterococcus in developed areas. So we have the septic tank leachate, we have waste treatment plants, we have also have injection well kind of development. So these are all sources that would lead to Enterococcus getting into the coastal waters. So let's get on to our motivation. So according to Defenders of Wildlife, they discovered that annually 8 million people visit the beaches, while generating $5 billion revenue. That tells us that the beaches are very, very important to the Texas state and also the US government. And so our hypothesis is that there is a link between pathology, hydroclimatic variables, and developed areas. So we intend to utilize machine learning techniques to understand the dynamics of pathology in species while creating a linkage with climate-induced events, population growth, and also man-made effects. And so after our meeting with our stakeholder, TGLO, and also looking at the data from the Texas Research Program online, we were able to narrow down these 4 counties as our area of interest because they have high levels of Enterococcus in these species in these counties. And so we decided to work with these counties in order to be able to understand the dynamics of Enterococcus in the species in these areas. And so I'll hand over to Chris to give us a walkthrough of our project. Thank you.
All right, so I'll be going over the work that we followed over the course of this study. So starting off with our primary objective, which is of course to identify some of these key drivers of Enterococcus within the Texas coastal waters. So in order to do that, we sourced various data sets from various ways that we will cover in the following slides. But some of these are data sets that have been used in previous studies to answer a similar question. And then some of these data sources are pretty novel, and especially with our cell phone data. So after we obtained all of our data sets, we transitioned into the data mining phase, which included us removing some of the NaN values that appeared within these different data sets, removing duplicates, things like that. And then the ultimate goal of this process was to create a master file to consolidate everything. It's just one central location. And so once that master file was created, we transitioned to our statistics and machine learning phase, where we created a principal component analysis to help kind of explain some of the variations between the variables and its relation to Enterococcus. And then we created different heat maps as well of our study area, and then ultimately our random forest model to help us identify the key drivers of Enterococcus. So that leads us to today, where we're happy to share some of our findings in terms of relations between some of these different factors within these data sets to Enterococcus levels, as well as what are some of the drivers that our random forest and statistics have pointed out as key drivers of Enterococcus. So I'll hand it over to Laura, and she'll begin discussing our different data sets.
Thank you, Quincy. So the main data set that we worked on for this project was Enterococcus bacteria that was sourced from Texas Beachwatch. And in our 4-county study site, there are 85 testing sites. And within the 2-year period of 2022 and 2023, there were 5,749 samples. As you can see in this map, a majority of the samples in studies or sampling sites do lie within Brazoria and Galveston County. Going on to our second data set, which is tides from NOAA. And this goes over water level, wind speed and gust, and wind direction, U and V. And within this, we were able to look at 10 monitoring sites in the 4-county study area. Moving on, we have our septic system data, and this is sourced from the Texas A&M University-Corpus and this is just GPS coordinates. It contains past and present, so it is difficult to say which ones are active and not active, but to address this concern, what we did was look at Google Earth, and if Google Earth said that in 2010, there was this neighborhood that was a little more spread apart, let's say each house was 2 acres,
And then in 2023, there are new suburbs in there, and the houses are cookie cutter and it's in city limits. We are going to assume that they are on city sewage and not on septic systems, and those septic systems were removed from this. And this map is what was left over after removing those certain neighborhoods. Going forward, we also have the potential source of contamination, PSOC from Texas Commission on Environmental Quality, and this is also just GPS coordinates. So what we mostly looked at was marina, municipal wastewater outfall, private wastewater outfall, and just wastewater. So this is just a GPS coordinate. There are no background results for how much outfall is occurring in this area, but we just like to have that in there to know what is in that surrounding environment. And then our last one, our fifth one, was cell phone density data. And this was sourced from the various movement product as from TGLO and HRI here at TAMU-CC. And the cell phone density data is supposed to represent the population density that's in the area and more so the anthropogenic part of this project. And what this does is this is an example of one day, and there is data for each day from March to August 2022, March to August 2023, and that is shown in our results as well. And this is an example of 24-hour period. Each little hexagon is 5 square miles, and these colors represent how many devices were in the area on that given day. So as you can see, a little bit red over here and red over there, which is using clearly. In that given 24 hours, there was between 3,000 to 5,000 cell phone devices. You can almost assume that's roughly about how many people were in that area on that day. And you can see most of the coastline is purple, so that means that less than 80 devices were picked up in that area. That's pretty okay, but that just gives us a rough estimate on how many people were there on the day. We chose May 3rd, 2022, because that is the most densest day in terms of cell phone devices in the state of Texas in our study area type. So I'm going to pass it off to Quincy to talk about our last variable.
All right, so our last variable that we included in this study is precipitation. So what we used were radar defined rain events for this study. And so we use the MRMS precipitation features database, which is courtesy of the ICON lab here at TAMU-CC. And what this data set is comprised of are rain events detected by a network of 160 NextRad radars that the National Weather Service utilizes. And each radar is capable within this data set of taking a scan every 6 minutes and at a spatial resolution of up to 1 kilometer by 1 kilometer. So we're able to get some pretty high resolution scans and snapshots of these rain events within this data set. So within our study area, we have a little over 300,000 unique rain events detected between the years 2022 and 2023. And each rain event is unique in the sense that it has its own location given by a coordinate. It has a rain rate attached to it in millimeters per hour, volumetric rain, so how much rain was released over the area, as well as the area of rain coverage, so how much area of the surface was receiving rainfall. So the figure on your right you're seeing is just a map of the volumetric rain contribution from the 300,000 rain events we have within our study area. And so the areas in red receive the most rain within our study period. And so the reason why this is important is that, of course, rain in itself is not a point source of bacteria, but it can be used as a transport mechanism for particles that are the point sources. And so for our area in particular, this area is very prone to hurricanes and tropical storms and just flooding events in general. And so you can have a scenario where you have this flooding rain event passing over a leaky septic system or a wastewater treatment facility or something like that. And then it's now washing these particles that should be contained within these septic areas into a nearby stream, which flows out into our study area, or it gets directly sent to the Texas coastal waters. So now I'll cover our master file that we utilize to conduct data analysis. So what we did was we took all those different variables from those previous data sets we just covered and then matched them to each individual sampling event within our study area. So all 5,000 events had a an analysis done to allow us to gain additional context of what is going on in the environment in the days leading up to the sample being collected. And so what we did was that we established a criteria spatially and temporally. So the spatial criteria that we used was 8 kilometers, which roughly translates to around 5 miles around each testing site. And so the 8 kilometers was chosen so that we could include some of the offshore rain events, as well as in the future when stream flow is added, you now have the, I guess, the ending points of these streams that flow into the Texas coastal waters. And then for the data that had a temporal component attached to it, such as the rain or the tides or the cell phone data, these variables to be determined as a match, they had to have occurred within that same 8 kilometer area, but they have to have occurred within 0 to upwards to 7 days prior to the sample being collected. So the figure on the right is just a visualization of what is going on in this matching process. So this is a real sampling event. So this was taken at Jetty Park #1 in Matagorda, Texas. And this site was sampled July 1st, 2020. So what you're seeing in the center is a black X, which represents the sample site. And then the different rings around it represent different distance thresholds, which I'll cover in more detail in the following slide. But all the other scatter marks that you're seeing are different sources that were detected near this site within the weeks leading up to this site being tested. So what you're seeing here are rain events denoted by the stars. And the stars are the colors are represented by the rain rate attached to them. And then there's also other anthropogenic sources that were identified to be associated with this sampling event. That included septic systems as well as wastewater sources. So what variables appear within our master file? So we have a little over 60 variables included on top of what Texas Beach Watch already records themselves. And so we broke them up into 4 different classes. So we have our rain class, PSOC variables, cell phone variables, as well as the tides and wind variables. So I'll define a few of these as they will pop up later on in our results section. But there's some variables that appear, such as rain 0 to 2 day, rain 4 to 5 day. So what these are telling us are the counts of rain events detected within the 0 to 2 days prior to the sample site, or whatever the date threshold is. And so the logic behind doing this is that we can, if there is indeed a correlation between rain and Enterococcus, we can start to pinpoint, you know, is it the rain event that is happening the day before sampling, the day of sampling, or could it be like the big rain event that happens, you know, 4 to 5 days out that has the most correlation with Enterococcus levels? Then there's some spatial variables as well. So we have rain 2 to 4 kilometers, for example, or we have septic 0 to 2, upwards to 6 to 8 kilometers. And then the logic behind these are similar in the sense that are they the sources that are closest to the testing site geographically that are influencing the Enterococcus levels the most, or are the ones a little bit further away? Then we have some mean values within some of our data sources, such as cell phone density 3-day mean, 5-day mean, 7-day mean. So what this is telling us is over the course of the 3 days prior, or 5 days, or 7 days prior to sampling, how many cell phones were within the area of the sampling site. And then within the rain variables, there is a mean volume rain and mean area rain. And so for all the rain events that were detected and determined to be matches to the sampling event, out of all of them, what is the mean volume rain released over that area, and what is the mean size of the rain event itself? So I'll hand it over to Avani, and he'll discuss some of our roadblocks and pre-processing techniques.
Thank you so much, Chrissy. Okay, so working on this project, we encountered some roadblocks, which one of these is choosing the right techniques to use the right machine learning model to use. So we try to go with random forest. And also the sample resolution of our data sets are very different. So that was a challenge for us. And then we had to address that as well. And then we had to transition to the address pro mapping software in order to have a good view or a good understanding of description of the variables and the interplay with the bacterial data and also on the study area. So moving forward, this is a GPM data that we initially acquired for the project. And looking at it, it's not really, it didn't capture the rain events at the study area very well. And then we had to transition to the ground-based radar, which got a very good outlook and has more detailed data about the rain events in the study area. And so moving forward, we'll look at the analysis and then look at statistics and machine learning results that we have for the project. So we ran a time series analysis using the weight variable and also the Enterococcus variable. And from each, you could see that the high levels of Enterococcus coincide with high levels of mean use speed at some instances. And that would say that the weight has a good effect, or I would say it's one of the drivers that we will have to focus on for this project, as it influences high levels of Enterococcus in the Texas beaches. I want to ask Laura who give us good insights into the variable index that we produced.
Thank you. So with the statistical analysis of the correlations that we had, we used the p-values of the top 10 most correlated variables to create a vulnerability index over the study area. And essentially what the top 10 variables consisted of were cell phone density, wind, volume, rain rate. So using those top 10 and across the study area, we just put it into our GIS with those p-values. And this is what it gave us, saying that kind of the area with Galveston city limits, so you can imagine where the gardens is, a high tourist area, kind of downtown, is where it was most vulnerable, along with the Surfside Freeport area. And that's not saying that the areas in the purple aren't susceptible to having a high Enterococcus level, but that these areas in particular, just based on their environmental conditions and what can happen there genetically, are more susceptible to having a higher level of Enterococcus. So moving on into the machine learning aspects of the project that we use random forest. And essentially, random forest is like a decision-making tree, and it can be used. So instead of just getting one result, we are able to get a variety and get a better outlook on everything that we were looking for in the bacteria data set. And random forest can be used in many different fields for land use changes, vegetation, and natural disaster risk areas like we used for the index. I'm going to pass it on to Quincy to talk about the results from our random forest model.
Thank you. So as Laura mentioned, we utilized the random forest model to conduct our machine learning method. So we use two types of random forest techniques. One method was a random forest regressor, and then the other was a random forest classifier. So I'll discuss the regressor first. So what this model was attempting to do was taking those 60 variables that I mentioned earlier, like the rain, septic, those variables, just only giving those can give us a value within plus or minus 20% of what was actually reported by Texas Beach Watch themselves. So Texas Beach Watch, whenever they process their samples, they classify them into 3 different modes. So one is safe, which is anything less than 33 colony forming units per 100 milliliters. Moderate, which is between 34 and 103 colony forming units, and beach advisor, which is anything greater than 104. So the model accuracy for the safe range, 69% of the time, whenever it encountered a true value that was within this range, it was able to accurately predict that the value was within this range. And then for moderate, 44% of the time, just only given those variables, it was able to give us a prediction. And then 31% of the time for the beach advisory category, it was able to give us a value within plus or minus 20% of what was actually reported. So I know that these percentages, they may not seem to be all that at the moment, but random forest models, they really thrive on having a lot of data. And so we're only using 2022 and 2023. And within those 2 years, we're only looking at 6 months worth of data in each to align with our cell phone data. And so we're confident that given that this is our baseline, that when we do acquire more data, that these results will improve. And so for the random forest classifier, this model, we're essentially being a little bit nicer to the model, in the sense where we're not judging it based on can you predict a value within that 20% range. We're just judging the model just based on just giving us variables. Can you tell us will it be anywhere within the safe range, anywhere within the moderate, or anywhere within the beach advisory range. So as you can see, the model accuracy did improve across all three categories. So 77% of the time, it was able to take those variables and classify a sampling event as safe. 65% of the time, it was able to take those variables and classify it as moderate. And then 51% of the time, it was able to classify it within the beach advisory range. And so, I guess another limitation besides the limited data is just that another limitation is just that we have a limited amount of samples within the moderate and beach advisory range in particular. So as mentioned earlier, we have a little over 5,000 samples total, and several thousand of those fall within the safe range, but only a few hundred fall within the moderate range. And so there are some techniques such as smoke, which allows you to artificially boost your data set and create, I guess, fake data in a sense, using what you already have to better train the models. And then, so I'll discuss the feature importances that both models output. So the left is showing the feature importance for the regressor, and then the right is showing the feature importance for the random forest classifier. And so this is just the model's way of telling us which variables it used most frequently to make its predictions. I forgot to mention previously, but the models are ran 50 times, and after those 50 times are ran, then we totaled up what was the most common variables within. And so these are just the top 15 model variables that the models use. There are some below this, but just for that space, we just chose these top 15. So the regressor model on the left, which was very interesting to us, it utilized wind speed and wind direction as very important variables, and wind U represents east to west winds. And then cell phone density was a very important variable, as well as septic systems were very important for this model. And so, as mentioned, this was a little bit of a surprise to us, and we were thinking about it, and similar to our rainfall, is that it's not a direct point source, but it can be used as a transport mechanism as well. And we conducted a PCA, which Afani will cover in the following slide, to kind of help explain this variation. And then on the right, these are the top 15 variables that the random forest classifier utilized. And so the top variables included mostly cell phone density, septic system, as well as area rain. And so what's interesting is that both of them did use wind as an important variable, but they used it in different levels of importance. So the regressor utilized wind as its top variable, whereas the classifier, although it did use wind, just wasn't as important. And another thing worth noting is that you can see in the classifier that you're a little bit nicer with a model's range, that it kind of expands its importance across the variables. And so the lengths of the bars are quite similar. Whereas for the regressor, there's a clear separation between the top 5 or 6 variables and those bottom 10 variables as well. So I'll hand it over to Bonnie to discuss our PCA to explain some of these variables.
Okay, thank you so much, Tracy. And so we took the 15 variables from the classifier that Chris already mentioned and then plugged it in the PCA. And what we did that because we use the density-based clustering algorithm because we are working on 4 counties as opposed to using the K-mean clustering. And so when we ran it, we were able to detect 3 clusters and then the noise. So the cluster 1 shows high rainfall and high septic influence. The cluster 2 shows moderate rainfall and low septic influence. And cluster 3 shows low septic influence and low sample influence as well. So overall, it's points to the fact that cluster 1, which is high rainfall and septic influence, is a key driver for the high levels of Enterococcus in the Texas beaches. I'll hand over to Laura, who will talk about nutrition that we encountered while working on this project.
So Quincy mentioned not having enough data, and we did limit ourselves in certain ways by using the cell phone density data, which is only for 2022 and 2023. If we were to use a different type, such as hotel tax residency, that could also give us another insight into how many people were in the area instead, and that could expand our data set to possibly 2017. But for this project, we decided to focus on cell phone density. Going forward, we could add another year of both Enterococcus bacteria for 2024 and cell phone density possibly. But as also Quincy mentioned, when you separate your Enterococcus levels into those concentrations of safe water, in beach advisory only has about 600 samples to work with 2 years, so it's different. But also with the addition of stream flow, that could also help. And then, as I mentioned previously, the septic system data and not knowing the difference between active and past ones was something that we tried to address, but it's not fully known in terms of data. Furthermore, going forward, obviously improving the model's accuracy would be one of the most important things. If there is a way to get it higher, and just by using different models that are out there, and then we would like to publish it. But I'll pass it over to.
So now that this project is concluded for us, or at least for this phase, within the next few days, we plan on finalizing our GitHub, where we will post our master file as well as some of our different Python scripts and techniques that we use for this study, so that someone in the future can continue on or just see what we did. So this is our final conceptual diagram to wrap everything up. So our statistics, what we found for the t-test, that the key drivers were cell phone density, volumetric rain and wind gusts, and then the AI models found that septic systems, cell phone density, once again, wind gust, wind direction, and rain coverage were important variables. So these are kind of broken into things that we can somewhat control and some that are totally uncontrollable. So things like wind and rain, we can't control those, but with further analysis and pinpointing, if it's indeed the rain events that are occurring several days out, or is it you have like a lot of septic systems or something like that leading up to the sampling occurring, then you can kind of use these uncontrollable variables to forecast the future and better inform people that are planning on going to the beach, just to let them know that, hey, we have this event that the past has led to high Enterococcus values, so just a heads up. So, and it was so funny to give us our acknowledgements.
Okay, thank you so much, UBC. And so we have come to the end of this project, and we're so happy to have completed this project. And we want to thank Stages for giving us this opportunity to be part of the traineeship program, and also thank our stakeholder, TGLO, and also thank our mentors that helped us as well, guiding us throughout this series projects. And most of that, thank also NSF for funding the Stages program. And with that, we have our references and have to take your questions. Thank you.
Great job, Mel. From your modeling that you've been able to do so far, how far in advance do you think you're going to be able to extend predictions now so that, to notify people? Have you got a good feel for how long timeline for that?
We haven't gotten too far yet, just based on the variables that we do have available to us in the short amount of time that we do have, the 2 years. And so we haven't really looked too far into using some of these variables as a forecasting technique, but we do kind of have a general idea with our PCA, which kind of explains, you have low wind, low rain, you have scenarios like that that could lead to an high Enterococcus value. We have done a little bit of analysis on things like that, but we haven't done 2 days out can you get a value close as possible to what is actually reported in nature? But that is something that we do plan on doing in the future, or data is available to us.
And then for the Enterococcus testing, it is collected in the summer months weekly. So a big part of our project was filling in those gaps and making the predictors between those weeks and the data right there.
Hi, great job. Okay, so I have a couple of questions, and you'll have to forgive my lack of this type of science knowledge because I'm a political scientist. I'm super excited about your data set and your modeling because I'm all about the human factor in this. So first, you mentioned that the cell phone density date that you picked was the most dense out of that year. Did I hear that correctly? Okay. Do you happen to know if it corresponded with things like spring break, for example?
That's a Tuesday. So I looked into it. And I'm like, why on earth would May 3rd a Tuesday? School is still in session.
Yeah.
And I personally could not find any news articles or events, and the Clear Lake area was dominant. Like if you were to look at 2022 and 2023 top 10 days, Clear Lake is about half of those. In terms of density, and they're well into the thousands. And I'm not sure what happens over there, whether that be prom,
or major the mega church gatherings, for example, or conferences. Okay, yeah. So because I think that's something policymakers would be really interested in knowing about. So speaking to Dr. Donaldson's question, right, as a predictor, like how many days out? And part of me is, of course, thinking about how there's a small but growing global movement to limit tourism or limit the number of people in a given area in concentrations, right? Because they know not just about the environmental damage, but also physical damage, right, that can happen in an area. But yeah, I think that's something that folks would be curious about for the work that you're doing. So maybe dipping a toe or two into the social science component of that, because that's a really powerful potential implication on the policy side of things. And then on a related note, I was wondering for not just the septic tank data, but I guess your general like water outflow data, did it include any information regarding the desalination plants that are in the area?
I'm not recalling the TCP having desal plants as a GPS.
Okay, yeah, I'm just curious about possible omitted variable bias, right? Like maybe there's something else that's going on with the waters in the area. And again, on the policy side, there may be implications moving forward, especially as Corpus Christi is headed in the direction, right? If I'm not mistaken, the first like seawater desalination, right? And so I'm wondering for our area, which is heavy tourism as well, like if that's going to be impactful in a quote good, bad, or not, a different kind of way for things like increases in bacteria. But yeah, great research. And then I think for the non-super scientists like yourself, like maybe when you do your GitHub, even a simple infographic might help. Like, so when you did your cluster analysis, which I think was really useful, I had really hard, and I was like, okay, is wind a smiley face or not? Like, is like not a lot of rainwater a smiley face or not? So even if you just if you get these three combinations together, you're more likely to get bacteria, bad, right? Red flag day. Because your general public who aren't specialists might get lost like in the numbers. And so then if you're thinking about like the politics of science communication, right, you have this fabulous data set, these great predictive models, but then at the end of the day, like, I don't know if I'm supposed to take my toddler to the beach or not, like, because of cooties. Right. So anything that y'all can do moving forward to help like your lay people, I think, process your data in the ways that still respect their knowledge and their understanding of general science, but also knowing like they're not going to know the machine learning like component of it. But yeah, great job.
Thank you for your
Yeah, excellent job, very thorough, well done, guys. On the variable importance, a couple of questions. One, did you repeat the variable importance and get kind of a sense of the variability for the respective variable importance. And then so we have people with the cell phones, we got wind, we've got rain, we've got septic systems. What is not there? I think water level are the other things that are not there. So the two questions. One, variability, and then the second one, what's not in your predictors, what we would infer from that.
Yeah, so in terms of water level, it is technically here, but like I said, we just clipped the top 15, so there are variables beneath here, like some things that are missing are marinas or wastewater sources as well that are here, but just not as important as we thought, because in previous studies they have shown that there is a linkage between water level and Enterococcus, and so that's why it was such a big surprise to us to see water level not being so high within the feature importance, but when instead. Sorry, can you repeat your second question?
So that's pretty interesting. That's why do you think water level, marinas, wastewater were not? Is it something that we can infer for the whole Texas coast, or do you think it's particular to the area around Galveston that you study?
Well, so one limitation as well for like those wastewater plants is that we don't have like their discharge data, we just have the coordinate, and so it's possible that one wastewater treatment facility could equate to 100 or 200 septic systems, and so that's just some things that we can't really dive too far into just given the nature of it.
And the other question was variability. When you rerun a random forest, it's going to give you different variable importance. It's just the nature of it. And so sometimes I repeat it the number of times and I get little error bars. And I was wondering if you did a little bit of that to get a sense of how stable. And pretty cool to have both the classifier and the regressor right next to each other. I think that gives a lot of confidence to the results.
Yeah, so what we did was we randomized the random state. So random state for random forest is like almost like a seed where you give it a number, and if you want to replicate what that model run did, you just give it that same exact number. And so what we did was we randomized the random state 50 different times to give it 50 different scenarios essentially. And so after those 50 runs were completed, then these were the main top variables that appeared across those 50 different random states.
So that's a mean value over 50. Yes. That's super cool. That's great. You should have added a little error bar to give the range for each of them. That's well done.
Okay, so it seems like the cell phone density, some of that wind is really important. Do you think those are like interconnected in some ways? I mean, I imagine it's really windy, maybe I don't want to go to the beach, so maybe on a low wind day, maybe go to the beach or something. Maybe tide and kind of move some pockets back here around. Do you guys kind of look at how those variables interact with each other?
Okay, so for the wind, it's very important because on a windy day, you expect high tides coming down, right? And so if there is a septic leaking system close by, it's possible that the wind can take the water and then gravity so inside close to waters. So the wind is actually very important as well. And then also looking at the fact that even during low wind period, the water is stagnant, and then that will give time for the bacteria to re-skim the water. So they are all very important. Let's see.
Hey, so you included septic system, which is human waste, but did you ever consider non-human waste, not birds, but agriculture, so cattle? Because a lot of your area was in regions where there's a lot of cattle, and so I was wondering if that was ever considered.
Well, actually, we didn't include that. I think it's something that we could consider going forward to really get to see influence in terms of agricultural increase whatsoever. So it's really good as well.
Thank you. I will add to Remy's question there. Remy, this is along the coast, along the Barrier Islands, so cattle might not be as much of an influence, but there are other sources, right, like birds and dogs, and so that's something pretty hard to, account for. But there are studies that are showing that they're coming up with the markers, right? It's the source tracking, so yeah, it's just difficult to get them in the models that are not necessarily available unless you walk the islands and you. Yes.
If you wanted the cattle numbers, the way the state health department does it is they go to the county ag agent and they get cattle numbers in every county. So it can be done. There's a fair number of cattle on the barrier islands, like on Matagorda Peninsula, they swim across, so they are there. But the other thing, there's some high numbers right along the Colorado. That area is infested with wild hogs, and AgriLife is doing some research there. So just kind of, if you ever were to go forward, you might consider that.
Thank you, Billy, to know that.
Yeah, we're very enthusiastic about your presentation, as you can see. I'd like to go back to Dr. Donaldson's question, but it seems like in this room there's probably enough experts. A frustration is bacteria. It takes 24 hours to know if the area is contaminated or not. And so your model is probably the first one that I see that would have a shot at doing predictions. If you wanted prediction just for tomorrow, you would need rain prediction. What else would you need to make it happen? And then we have quite a few experts in the group that could give you an assessment if that would be possible, and that's like predictions, prediction. What would you need to make a prediction for tomorrow?
At least as of right now, with the model that we have, well, septic system and the potential sources of contamination, those aren't going anywhere. Those are just GPS coordinates. Precipitation and the tidal data from NOAA, those would be the two most accessible ones that we would need today to get it. The cell phone density, there's ways around that. You can try and go by patterns because that's not the most accessible data to get. So just going by what events are happening in the area in the past, how many people have to the beaches in that particular time for this, and make an estimate from possibly how many people are there. Could do that, but the most, I guess, time immediate ones would be precipitation, and then the NOAA tidal data. So water level, and then mostly the wind.
Water level data, we got that.
Yeah.
Precipitation, they use the.
It's an add-on to the wind, in terms of wind directions. I mentioned that wind yield direction was a pretty important variable. So going back to wind being used as a transport mechanism, and it could be related to current direction. You could possibly have a known source of contamination, coming from the southeast or something like that. And then you have a strong prevailing wind from the southeast, but around the site, the immediate area around the site, you have low rain, or you don't have variables that are indicative of a high Enterococcus volume. And so you could possibly begin to backtrack from where the wind direction is coming from and identify what sources are from that direction as well.
So I noticed that when you do the regression, east-west wind is more important than south wind. But when you look at specification, your north-south wind is really comparable to all other variables. And I would say, depending on the rain system coming in, if it's in the springtime or front coming in, they have a pretty good prediction in 7 days. We'll see. In the summertime, for the thunderstorms, many times we have a little bit low confidence on those thunderstorm happening, with a shorter prediction, but I think we have pretty good understanding of the wind and the precipitation, and for the it's really different, you know, the volumes. But any comments on the way against these last rains, the predictors?
It's similar to East West, just as we have a possible scenario where you have various sources coming from the south compared to the north that could possibly be playing a role. And then as mentioned for the regressor, this is, if you look at the entire list, past 15, a lot of the bars are very similar, and so it's possible that wind yield is important variable number 16. So it's kind of hard to differentiate between why north, south, east, west are hearing differently between the two models, but it could just be the nature of the classifier itself.
Is that daily rate? Daily means?
The wind.
Yeah.
Yeah, so the wind V that you're seeing down there is over the 5 days prior to sampling. What was the average wind direction or predominant wind for those 5 days prior?
Follow-up question to know, or? Well, you're the last group, so you're gonna take a lot of the questions.
Thank you for your job. I don't know, this question is very stupid question. I wonder. I believe you use a p-value is 0.05 when you do this test. But I wonder, you use predicted value, in this screen, you use predicted value within 20% of actual value. So do you have any specific reason to use 20% actual within the 20% actual value?
So after talking with Texas General Land Office and Texas Beach Watch, that value that they report is has high variability, and so one example that they give us is that if we go out into the water, let's say 5 of us, and we all sample the water at the same time, that Enterococcus value that's reported can vary greatly across all 5 of us. And so just to give us a way to kind of explain this variability, we just chose 20%, but it wasn't like a statistical method to get this number, we just kind of picked it.
I can briefly add to that. What Quincy is saying is that the measurements, the actual measurements, not only that, you know, you might have bacteria here, but not have it 5 meters away from where you are. So there is a very large error, per se, in those measurements. And when you're trying to predict actual values reported, you're going to have a hard time, right? And yes, and so then too, with the winds, we have to remember that there is dilution, that if the wind is towards the shore, there's going to be offshore waters that are, you know, have less bacteria, versus if you have the wind blowing away from the shore, that's going to pull some of that, either stream flow that's coming contaminated or from the subsurface. So there is a lot of, it's a very dynamic environment, and it's chemistry. It's not as easy to predict, and it's not a linear relationship either. So just so we wrap it up, because we are getting close to lunch, and I really appreciate everybody's input, and we're going to continue the conversation for sure with the next cohort if they choose to continue on this project or any of the projects. I just want to remind everybody that our trainees are doing these projects on top of everything else they're doing for their actual degrees and coursework. And so there isn't enough time to do it all and play with these models, but we're hoping that maybe the next cohort is being implemented, that our stakeholders are seeing a little bit of that light at the end of the tunnel, and we can engage our researchers to work on the group projects with them. So it shouldn't stop here. It doesn't stop here. So please feel free to mingle, and lunch is gonna be served. I am being told that at 12:20, there's gonna be the mariachi band playing next door. So you could go and enjoy that quickly. You just don't know, we're just right next door and it's not there. So, and we resume at 1. Okay, thank you so much. Thank you all.